Okay, great. Thank you, Megan, for the introduction, and I'd like to just thank the organizers of the symposium. I'm really delighted today to have the opportunity to talk about how we're leveraging untargeted high-resolution mass spectrometry approaches for investigating complex exposures in humans. We had a fantastic presentation by Dr. Lee Ferguson right before mine, discussing kind of the nuances and difficulties in computational approaches for processing high resolution mass spectrometry data. So I'm going to really focus on the need to use these methods to study exposures in humans and how we're leveraging them in new studies that aim to investigate complex but relatively uncharacterized exposures and their health effects. So I'll begin my talk by just describing the critical role for using untargeted assays in characterizing the human exposome. And then I'll follow that up with a few examples of how we're incorporating these untargeted assays to study emerging environmental health concerns. These include micro and nanoplastics, as well as many of the perfluorinated chemicals that we know are being used in industrial applications, but may not have been specifically characterized or examined in human populations. So, many of us as environmental health professionals know that genetics alone does not describe our risk for disease. There's been a lot of emphasis on using a genome or genetic-centric perspective of disease risk over the past 20 years. And genetics has provided a significant amount of important insight into understanding what can drive disease risk. However, this isn't the whole story. Our current estimates suggest that about 5 to 15% of diseases can be attributed to heritability alone, and the remaining 85 to 95% are derived from environment, lifestyle, and their interaction with the genome. So therefore, if we are only considering heritability or genetics in human health and disease, this is really an unbalanced perspective that we're taking. Unfortunately, there's a discrepancy between our ability to characterize the environment relative to biology. So there's been tremendous advances in systems biology and omic technologies that allow us to characterize multiple levels or profiles within a human sample. These include things like whole genome sequencing and genotyping and transcriptomics. We can look at epigenetics and methylation, as well as proteomics. Unfortunately, our ability to characterize the environment, even using some of the best technologies that we currently have available, hasn't really seen the explosive growth in capabilities that we saw in other omic sciences. We know the potential for the exposome is huge, and chemical space can encompass millions and millions of compounds, which could inform our exposure history over our lifetime. And so because of this, there's a critical need for us to develop analytical frameworks that allow us to perform universal screening of the human exposome. The way that this has been approached in both my laboratory and many others that are trying to develop new methods for measuring the human exposome is through developing analytical strategies that allow us to characterize the human metabolome. The human metabolome can be defined as the complete collection of small molecule metabolites found in the human body, and it is this definition that makes it such a critical measure for exposome research. Within the exposome, or within the metabolome, we have the capability of screening for environmental chemicals, commercial products, and supplements and pharmaceuticals. And these represent key measures of our exposure within the exposome concept. At the same time, within the metabolomic measures, we have the capability of characterizing alterations in endogenous pathways and how those are linked to both exposure and disease. So it gives us the ability to characterize biological response and effect within the metabolome. So this measure, by providing a central link between exposure through detection of internal dose or exposure biomarkers, as well as biological response through detection and alterations in endogenous pathways, provides a critical capability for characterizing the exposome in human populations. Now, as mentioned before, we know that the exposome is considerably larger than that we can measure using traditional analytical approaches based upon targeted analysis. And so to truly measure the exposome, the analytical methods that we develop need to expand beyond traditional approaches that are used for biomonitoring specific levels of environmental exposures. And to do this, we incorporate what's referred to as untargeted chemical profiling techniques. And these differ conceptually from traditional analytical approaches in that the objective of this analysis is to maximize the number of compounds that you're detecting. And we're using this to identify the exposures that could be driving potential health effects. This is very different than what we would use in a targeted or biomonitoring study where we're identifying certain exposures a priori, developing methods to measure those, identifying populations that we can test hypotheses about, or using those exposure measures and identify exposures linked with outcomes or different diseases based upon that prior identified set of compounds. In untargeted chemical profiling, we're maximizing our ability to detect exposures as well as endogenous metabolites oftentimes, and then using bioinformatics and biostatistics to determine which exposures we should be prioritizing for their relationship with their outcome. And I think that this is a critical recognition that we need for advancing exposome science. And that is to truly understand and embrace the complexity of the human exposome. It's critical that we adopt analytical strategies and study designs that incorporate untargeted measures of exposure. The EPA alone has over 80,000 chemicals that are registered for use in commercial applications. This doesn't consider the many abiotic and biotic transformation products that can arise from these compounds. So we need analytical techniques and strategies that allow us to measure exposures that are greatly beyond the scale of targeted approaches. And one thing that I found really interesting is that we, tend to think about these untargeted methods as cutting edge or, this new way to do environmental health research. But really, even some of the most traditional exposures or persistent exposures that we've been studying for decades, were really initially identified using untargeted methods to characterize biological samples. In this case, they were tissue samples from birds and other species. And so, I just want to take us on a quick history lesson here and describe the PCB story. And this was the work of an analytical chemist named Søren Jensen in Sweden, who was really one of the first to identify the presence of polychlorinated biphenyls in tissue samples, back in the early 60s. So this chemist, Søren Jensen, what his initial research was focused on measuring DDT and DDE metabolites in tissues from birds that they were finding deceased in Sweden. And so what I'm showing here on the right is one of the chromatograms from his initial analysis of these tissue samples, again, where he was aiming to detect and quantify DDT and related metabolites to see what the levels were in these birds. And when he did his analysis, he found that he was able to, very distinctly and uniquely detect peaks that corresponded to DDT and DDE. But what was interesting was when he analyzed multiple samples from different samples from multiple carcasses and species, he was repeatedly finding these peaks that were alluding at later retention times that weren't necessarily corresponding to DDT and DDE, but he was finding them in a number of these samples. And what's interesting to mention here is that these were just peaks. They were unknowns. This was not being measured using a mass spectrometer. This was an electron capture device. So, there was some suspicion that these were chlorinated compounds, but he couldn't, unambiguously identify what these were. These were truly unknown. These were unknowns in the truest sense. And so as he continued analyzing samples, he noticed a number of interesting patterns in the different types of samples he was analyzing. So the results on the left show the increasing intensity of these unknown peaks as you move up the food chain. So obviously the pike, which is a fish, eats young salmon, and then the white-tailed eagle will eat the pike and other aquatic species. And so what this is telling us is that these compounds are bioaccumulating up the food chain. So it's a relatively, there is exposure that's occurring across the food chain, but as the animals at the top of the food chain are eating these lower species, they're essentially accumulating levels of these compounds up the food chain. But then if you look at other species that are not carnivores and are eating grass and other vegetable-based diets or vegetarian-based diets, you do not see accumulation of these compounds. So this chromatogram here is from an elk. You can see that DDT, DDD, and other organochlorine pesticides are present. These are organochlorine pesticides that are being applied to plants and to prevent insecticide growth. So it makes sense that accumulation would occur in primarily grass-eating species. But again, we do not see accumulation of these unknown peaks within herbivores. And so finally, there was a sample that was found to be extremely highly contaminated with these unknown peaks, to the point that the levels were high enough where he could take an extract and run it on a relatively new technology referred to as mass spectrometry. And using the spectra he obtained from the mass spectrometer, he was able to identify these compounds as polychlorinated biphenyls. He purchased a commercial mixture of these compounds and ran it on the same instrument, was able to match retention times and spectra, and and conclusively prove that those peaks did in fact correspond to PCBs. And I'm sure, as many of you know, these are some of the most widespread persistent organic pollutants in the world. They've been linked to a number of health outcomes and have been studied, probably more than any other persistent organic pollutant to date. But what's really critical, and the take-home message from this, is that these environmental exposures that we all know so well today were originally discovered using untargeted methods based upon complete unknowns. Again, he was not able to initially assign a structure or a mass or any additional information to these peaks. It was purely a peak at a given retention time that was being measured reproducibly. And I think that this is so important for us to remember as we're adapting our untargeted methods to study exposures in humans, is that we have to remember, that while it is good to have as much evidence as possible identifying what we're measuring, there is also importance in considering these compounds that are unknowns and can be reproducibly measured. So I think it's always important to keep that in mind as we develop new standards and approaches for our untargeted methods. And so, as many of you are aware, and as Lee did an excellent job showing in his presentation, high-resolution mass spectrometry has really become a critical platform for untargeted methods. It allows us to do very sensitive and selective detection of a very wide range of compounds, and it also provides the capabilities to predict potential formulas and identifications of these compounds. I say predict because, as always, we need to have some evidence supporting what our identification is based upon. An application of these methods through exposome research has really shown the power of using them. We've been able to greatly expand our detection and measurement of unknown and undocumented chemical exposures. We've identified, new pathways of biological response that we can link to those exposures. And then we've also started developing approaches that allow us to characterize exposure memory. So what are the long-term metabolic changes associated with exposure to compounds? And so there's really been, huge advances in these approaches that have informed us on exposures and their effect. And so the question is how can we continue to leverage these untargeted assays to really push the cutting edge of exposures that we're characterizing? A lot of the work in my research laboratory has been focused on taking these high-resolution mass spectrometry methods and adapting them to measure very complex, heterogeneous exposures in humans. And I think these approaches are very well suited for measurements of things like microplastics, which are not a single class of compounds, but can represent a very complex and different exposures depending on the transport or origin of the plastic. And so microplastics are small plastic particles that are generated from a number of different processes. They've been detected in food and animals and in our air. And we think there is a relatively high amount of ongoing exposure to these particles. There's also been a lot of attention of the long-term transport and measurement of these plastic particles, both in environmental and initial human samples. The human work is very preliminary. There's been studies showing that these particles are atmospherically transported, and they've been measured in what's considered pristine mountain catchments. They've been measured in both raw and treated drinking water. They've been measured in seafood and other prepared foods. There's been some initial studies showing that they can be detected in biological samples, including feces and placenta. There's been some findings show that small plastic particles have been linked to different lung diseases. And finally, they've been measured in pet food and feces from a number of different sources. And so I think, a lot of what we're focusing on is leveraging what we consider an exposomic framework for understanding the potential health effects of microplastic exposure. And by what I mean by an exposomic framework is we're leveraging the strengths of the untargeted high-resolution mass spectrometry approaches to provide in-depth characterization of exposure to microplastics, as well as using more traditional approaches to better understand certain characteristics of the particle. So for example, we're using untargeted GC high-resolution mass spectrometry with pyrolysis for quantification of particles. We're using Raman and other microscopy approaches to understand morphology. We're looking at spatial mass spectrometry approaches to study compartmentalization. So how do these particles preferentially accumulate in different areas of tissues? And also untargeted small molecule profiling for mobilization. So what are some compounds that are being leached or are attached to these particles? And finally, we're linking that to biological response measures to see potential health effects of these particles. And within this, untargeted high-resolution mass spectrometry has provided a critical measurement for different aspects of the particle. And what I'm showing here is work that's been done by a research associate in my lab, Zoe Cotes. And so, again, I mentioned we've been using pyrolysis high-resolution mass spectrometry, and that allows us to quantify and screen the particles. We're looking at using a depolymerization method to look at additives and other particles. As well as using small molecule profiling to measure absorbed and co-transported contaminants. And we're using these methods and incorporating them into one of the first studies of the effects of microplastic exposures in humans and to evaluate some of the risk of these particles. And these include methods where we'll provide the first deep characterization of microplastic particles in human populations, as well as develop scalable high throughput methods that allow us to biomonitor for microplastic exposures in humans. We're combining that with toxicological approaches or studies in human populations to see what the risks are of these particles and how they're linked to different outcomes. And our hope is that we'll essentially develop a framework for risk assessment of microplastics. We're performing exposure assessment to see levels and distribution of these particles. We're performing toxicological studies for hazard identification, and then we're incorporating this within a general risk characterization framework to hopefully provide recommendations for minimizing and understanding microplastic exposures. Additional work includes perfluoronics, where we're using untargeted methods to look for the distribution of perfluorinated compounds in biological samples. So we're expanding beyond the traditional 10 to 20 that have been measured in targeted assays to try and screen for as many of the commercially used PFAS as possible. We've been able to show that we can detect a number of these in children beyond those traditional PFAS, including as well as the traditional perfluorinated chemicals using untargeted methods. This is very important because it shows that we have both reproducible and sensitive measures of the things we're interested in, as well as being able to detect other species. And we can also use the ability to detect endogenous metabolites to perform essentially a biological prioritization of which of these PFCs are important to study. And this is an example of how we're integrating perfluorinated compounds with alterations in the metabolome and seeing how those are linked to different pathway alterations that we know are consistent with disease processes. So I'm sorry, I went a little bit over, so I'll summarize these very quickly. But the conclusions, are that untargeted assays based upon high-resolution mass spectrometry provide a key tool for exposome research and a powerful basis for developing new approaches to study complex exposures. We're leveraging these approaches to measure micro and nanoplastic particles in human exposures and hope to provide new insight into the distribution and levels of those particles in human populations. And then we can combine untargeted methods with suspect screening analysis to look for very large or broad classes of exposures and characterize those relative to health outcomes. All of this work has been supported by a number of collaborators and funding from NIH and the European Commission. These are collaborators both at Mount Sinai and Utrecht in the Netherlands, as well as Emory University. And just before I end, I'd like to make a quick plug for the brand new Exposome journal that was just publicly released yesterday, actually. We're hoping this will be a home for articles that have an exposome focus, from pushing the technology forward to application of exposome methods to both human and toxicological studies. So as you're trying to find journals to submit your articles to, I highly recommend considering this journal in the future. And thank you, and I apologize for going over by a few minutes.